Good morning, all of you. My name is Praveen Nandevchan. I am from Jalna. I recently completed my B.Pharm, D. Batu University. Today's subject is CDM and CRA, and today's topic is what is clinical research. The first of all, we see the introduction of clinical research. Clinical clinical trial is a systemic investigation in human subject for evaluating the safety and efficacy of new drug. Clinical trial are the set of tests in medical research and drug development that generate the safety efficacy data for health and intervention in human beings. Introduction of clinical trial conducted only when the satisfaction information has been gathered on the quality of the non-clinical safety. The health authorized ethics committee approval is the granted. The country where approval of the drug is site. The clinical trial is the ministry for bringing out new drug to the market. Then we will see drug drug review steps. Preclinical testing. This is done in animal. Then number two, an investigational new drug application IND. Then phase 1, phase 2 studies, then phase 3 studies, then submission of new drug application NDA to the formal step asking the FDA the consider a drug for marketing approval. FDA reviews will approve the application or find it either approvable or not approvable. And then phase 4 is last study, post marketing surveillance, drug review. Before one can initiate the testing in human beings, the expensive preclinical or laboratory research is required. Research usually involves years experiment in the animal and human cell. If this stage of testing is successfully, the sponsor then providing data to the FDA requesting approval being testing in humans. This is called as investigational new drug application. The approved by the FDA, the testing of human beings, this done through the formally written and approved protocol. Then preclinical evaluation phase, animal studies, major areas, the pharmacovigilance studies in vivo, animal and in vitro preparation, the absorption of distribution elimination study, pharmacokinetics, acute, subacute, chronic toxicity studies, toxicity profile, therapeutic index safety and efficacy evaluation. Then IND application filling. One preclinical study have the indicated the safety and the efficacy of the drug. IND application has to filled with the regulatory authorities for the obtaining regulatory approval for phase 1, phase 2, phase 3 clinical evaluation. The contents of IND application, investigational new drug, preclinical data, all information on composition and source of drug, then chemical and manufacturing information, then proposed clinical plans and protocols, then ethics and committee clearance, phase of clinical trials. Then we will see phase of clinical trials, discover science, study in the lab, then study in the animal models, then phase 0, first study in a patient to the see how body metabolizes the drug. Phase 1, study a small number of patients to the evaluate safety and dosing. Phase 2, study of more patient assessment drugs effectiveness and further evaluation safety. Phase 3, the study with the largest number of patients, confirm drug is effective and safe, access side effect and compare the drug other current treatments. Phase 0, study of microdosing, study study of new drug in microdose to drive pharmacokinetic information. Human beforming undertaking phase 1 study is called as phase 0, microdose, less than 1/100 dose of the test substance, calculated procedure, pharmacological effect with the max dose greater than 100 microorganism. Objective to obtain preliminary pharmacokinetic data, preclinical data, subject acute toxicity study in one species by two routes of administration. Then phase 0 study, microdosing advantage, less chance of adverse effect, short duration, less number of volunteers, reduce the cost of development, reduce the drug development time. Limitations, study mainly based on pharmacokinetic parameter, non efficacy and safety based. Then we will see phase 1, first stage of testing human subject, designed by access safety laboratory, pharmacokinetic and pharmacodynamic drug. There are 20 to 80 healthy volunteer, duration of 6 to 12 months. The patient anti-cancer drug AIDS therapy aims. The phase 1 trial determine the maximum tolerated dose. The new treatment kind phase 1. SAD, single ascending dose studies, multiple ascending dose studies, food effect, etc. Then we will see phase 2. The therapeutic exploratory trial consists of 80 to 300 patients, healthy volunteers, to confirm the effectiveness of monitor side effect, further evaluatory safety. First inpatient, duration of 6 months to several years. Then we will see phase 2, the new action marketed drug with the phase 2. Phase 2 is the types designed access dosing requirement. Phase 2 design study efficacy, clinical trials. Then we will see phase 3, therapeutic confirmatory trial, target population 100 to 3000 patient, duration taking long up to 5 years, establishing efficacy of drug against the therapy, larger number of patients, method users collect safety data, etc. Then we will see end of clinical trial activities. The sponsor expert committee review the efficacy, safety and potential scales. Go number of decision files, new drug application, DCGI, expert review, DCGI committee, DCGI approval in CC marketed. Phase 4 begins. Phase 4 is post marketing surveillance. Okay, thank you.